In our last unit, we learned the rules of inheritance and that genes encode the properties of an organism. We also learned a little bit about the physical nature of the gene, that it's a discrete sequence of DNA along a chromosome. We've already learned that genes encode the properties of an organism by encoding proteins. In this unit, we'll learn exactly how genes encode proteins. Much of this unit will therefore be spent understanding how a sequence of DNA is turned into a protein. Before we get into that, however, I will remind you of something we learned early in the semester. DNA is the genetic material. This illustration tells us how we know. The experiment described here is different than the one described in the Campbell textbook. They discussed the Hershey-Chase experiment as evidence that DNA is the genetic material, and it does indeed show that. But this experiment was done first by almost a decade and is more definitive in my opinion. The experiment depicted was first done by Frederick Griffith. Oswald Avery built upon that earlier work to show that DNA is the genetic material, and he published his results in 1944. It's a complicated experiment, so let me walk you through it. We start with a strain of bacteria that is non-virulent. That is, it doesn't cause disease. We call this strain R because it has a rough appearance at its surface. We inject it into mice, and the mice survive just fine. It's non-virulent. Compare this to a similar bacteria, the S strain, S for smooth, which is virulent. When you inject mice with the S strain, it kills the mice. Virulence. You can heat up the S cells and kill them. That's one of the tools we have for killing bacteria, heat. It's one reason that we cook our food. If you heat kill S bacteria, it is no longer virulent. You can inject it into the mice, and the mice survive just fine. Here's the really interesting part. In the last experiment, they injected a combination of heat-killed S cells and R cells, two non-virulent strains, and the combination killed the mice. Not only did the mice die, but you could recover live S bacteria from the dead mice. What's happening? First of all, let's be clear that the dead S cells are not magically resurrected. Dead cells remain dead. Rather, while the S cells are dead, their cells are still more or less intact. So the interpretation is that information is traveling from the dead S cells to the live R cells and reprogramming those non-virulent R cells to virulent S cells. This process is called transformation, as in the phenotype of the bacteria is transformed from non-virulent to virulent. This is where Avery picks up on the experiments, and he asks, what is the transforming principle? Or, as we might say it today, what is the genetic material? And he was the one who first defined the criteria for the genetic material, that it transfers information from generation to generation, and that it encodes the properties of an organism. I won't go into the details, but through a rigorous series of experiments, he discovered the answer to be DNA, and decidedly not protein. Avery and his colleagues, building on the earlier work of Griffith and his colleagues, showed that DNA is the genetic material. So let's spell out exactly what we mean when we say that DNA is the genetic material. Genetic material has two properties. It encodes the properties of an organism. That is, a species is what it is, different from other species, because of its DNA. Specifically, the sequence of its genes. DNA encodes the properties of an organism by encoding protein, and it encodes proteins through the processes of transcription and translation. It also passes information on to subsequent generation. If a species is adapted to its environment, it wants to keep the traits that make it well adapted generation after generation. Information about those traits is passed on in the form of DNA through the process of replication. So when we talk about molecular biology of the gene, we're really talking about the processes of transcription and translation and replication, and that's what we're going to learn here. That brings us to the central dogma of molecular biology, which states that DNA encodes RNA and RNA encodes proteins. That's the central dogma in its simplest form, but let's unpack it a little. The central dogma is really about the flow of information. Information flows from generation to generation and from DNA to protein. Information flow to subsequent generations is necessary to maintain a species. DNA accomplishes this through replication, production of two identical DNA molecules from one starting molecule. In replication, DNA serves as a template for the production of more DNA. That's what the circular arrow at the top is meant to depict. DNA encodes properties by encoding protein. The process by which the code in the sequence of DNA leads to a protein is called gene expression. It includes the processes of transcription and translation. In transcription, DNA serves as a template for the production of RNA. In translation, RNA serves as a template for the production of protein. Now that we have the big picture of information flow in mind, let's get into the nitty-gritty and talk about nucleotides. Nucleotides are the monomer subunits that make up the polymer nucleic acid. By nucleic acid, I mean DNA and RNA, which mediate the transfer of information. Like carbohydrates are polymers of simple sugars and proteins are polymers of amino acids, nucleic acids are polymers of nucleotides. Three functional groups make up a nucleotide: a ribose sugar, a nitrogenous base, and a phosphate group. You might recognize this molecule. It's similar to ATP, which we've talked about a lot in the past. ATP is a triphosphate. This is a monophosphate, and this is a guanosine, not adenosine. The sugar group is a ribose sugar. That is a 5-carbon sugar, not the 6-carbon sugar of glucose. Finally is the nitrogenous base, so named because it has a lot of nitrogen and it's basic. While the nitrogenous base is basic, this phosphate group is very acidic, making the molecule overall acidic. That's where we get the word nucleic acid. They are acidic molecules present in the nucleus of cells. Nucleotides are very important in DNA replication and gene expression, so let's take a closer look. Like amino acids, there are different nucleotides that make up nucleic acid, so let's examine the parts of a nucleotide. The phosphate group is the same from one nucleotide to the next, and we don't need to consider it further. The nitrogenous base comes in 5 different forms: adenine, guanine, cytosine, thymine, and uracil. But you don't have to remember all that. You can just say A, G, C, T, and U. 3 of these bases, A, C, and G, are part of both DNA and RNA. T is just in DNA, and U is just in RNA. There are also different forms of the ribose sugar, ribose and deoxyribose. You can see the difference, which is an oxygen attached to the number 2 carbon. Deoxyribose doesn't have that oxygen, hence the name deoxyribose. That's also where we get the name ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. Note that the carbons on the ribose are numbered, and this numbering holds for the complete nucleotide. The phosphate group is bound to the 5' carbon, and the 3' carbon has a hydroxyl group. So the nucleotide has an orientation, a 5' end and a 3' end, just like proteins have an N terminus and a C terminus. As we'll see, that orientation carries through to RNA and DNA. This shows 2 differences between RNA and DNA. RNA uses ribose while DNA uses deoxyribose, and RNA uses uracil while DNA uses thymine. There's a third difference that's not on this slide. RNA is single-stranded and DNA is double-stranded, as we'll see in a moment. Now the question is, how do these nucleotides come together to make a nucleic acid? We'll concentrate on DNA instead of RNA because it's a more interesting structure. That structure was famously solved by Watson and Crick in 1953. They're shown on the right with their famous model of DNA. It should be noted that Watson and Crick built their model based on data generated by Rosalind Franklin, shown on the left, with the X-ray crystallography that ultimately led to the structure of DNA. Here's another model of DNA that's a little bit more convenient for us to look at. Let's start on the left, and you can see that DNA is made of 2 strands. Each strand is a polymer of nucleotides, a polynucleotide, held together by the covalent bonds of the sugar-phosphate backbone. Sugar-phosphate, sugar-phosphate, sugar-phosphate. The enzyme that puts together these nucleotides into a polynucleotide is called DNA polymerase. Note that 2 polynucleotide strands make a DNA molecule, and each one of them has a 5' to 3' orientation. The strand on the left is 5' to 3' top to bottom, and the strand on the right is 5' to 3' bottom to top. The strands run in opposite direction, or they're anti-parallel. The strands are oriented with their sugar phosphate backbones on the outside and the nitrogenous bases on the inside, which can now interact with one another. Hydrogen bonds between the nitrogenous bases hold the two strands of DNA together. In this case, we'll call them base pairs because they form between the nitrogenous bases of the nucleotides. Base pairs are very specific, and you have to learn the base pairing rules. But don't worry, they're easy. Adenine binds with thymine, AT, and guanine base pairs with cytosine, G to C. Or we could say that A is complementary to T and G is complementary to C. One strand of DNA is complementary to another. Base pairs, abbreviated BP, are also the unit we use to measure the size of DNA. The molecule depicted here is 1, 2, 3, 4 base pairs long. A typical chromosome in a human cell is millions or even hundreds of millions of base pairs long. Hydrogen bonds, as we know, are weak, but string millions of them together and you get a very stable double-stranded DNA molecule. The double helix is just that, helical, not flat as depicted in the picture to the left. A more accurate depiction is the double helix at right. The thick blue lines on the outside represent the anti-parallel sugar phosphate backbones, and, once again, the nitrogenous bases are on the inside, forming base pairs. DNA is a right-handed helix. If you twist your right hand around the helix, your hand will move in the direction of your thumb. If you twist your left hand around the helix, your hand will move in the direction opposite your thumb. Dimensions of the helix are on the diagram if you're interested. Watson and Crick published their structure in 1953 and won the Nobel Prize in 1963. Rosalind Franklin died of ovarian cancer in 1958 and did not share in the Nobel Prize. It's possible that exposure to X-rays contributed to her cancer. When Watson and Crick published their paper in 1953, they included this famous quote toward the end. It has not escaped our notice that the specific pairing that we have postulated immediately suggests a possible copying mechanism for the genetic material. In other words, the structure of DNA all by itself suggests a mechanism for replication. Replication is making two identical molecules of DNA from one initial molecule. And for this class, that's what you need to know, the structural basis for DNA replication. In a molecular biology course, you would need to know the detailed mechanism and all the enzymes involved. We'll get into mechanism a little bit, but mostly make sure you understand the structural basis of DNA replication. And remember that DNA replication is how information passes from generation to generation. Here's the simple description. DNA is a double helix held together by hydrogen bonds, or base pairs. Because hydrogen bonds are weak, the strands can be separated, and each strand individually can serve as a template for the synthesis of the other strand. This is possible because of base pairing. Where the template strand has an A, the new strand will incorporate its complement, T. Where the template strand has a G, the new strand will incorporate its complement, C, and so on. Eventually, each strand is completely copied, and we have two new identical molecules of DNA. When we consider the details of DNA synthesis, a complication arises. DNA is only synthesized in the 5' to 3' direction. Note that in one case, synthesis of the new strand is continuous, and in the other case, it's discontinuous. We'll see why that is in a moment. The other thing to notice is that replication is semi-conservative. That is, the daughter molecules that result from replication each contain one newly synthesized strand and one parent strand. Now let's look at a real-world example of DNA replication, inside a human cell, for example. The human genome is big, very big, 3 billion base pairs. If you were to replicate that DNA one chromosome at a time, it would take over a month to replicate. But of course, the DNA isn't replicated one chromosome at a time. all the chromosomes are replicated at once. But it goes further than that. Replication of each chromosome begins at many sites, origins of replication. That's what's depicted at left, many origins of replication along a single chromosome. Here you can see replication beginning at several sites along a chromosome going in two directions. These replication bubbles will continue in both directions until they meet each other and replication is complete. One half of a replication bubble is a replication fork, and it illustrates nicely the problem I alluded to before. DNA polymerase can only synthesize DNA in the 5' to 3' direction. This is not a problem for one of the strands, the leading strand, which is on the bottom in this case. Synthesis proceeds in the fork as it opens. The other strand, the lagging strand, has a problem. For the upper strand, 5' to 3' synthesis means starting at the fork and coming out. As the fork opens up, synthesis has to start again and again. Consequently, DNA synthesis is discontinuous. Small fragments of DNA are made as the fork opens up. These are called Okazaki fragments after the man who discovered them. The small Okazaki fragments are stitched together by another enzyme, DNA ligase. One more thing that's worth saying is that DNA replication is subject to occasional mistakes, and the cell has an array of repair mechanisms to ensure replication with high fidelity.